DNA replication. You know that DNA is the genetic material and how it replicates. We will be studying this under this topic. Three probable methods have been suggested for the replication of the DNA. They are conservative, dispersive, and semi-conservative. We do not have any evidence to prove that DNA replicates in dispersive or conservative mode of replication, but we do have evidence for the semi-conservative mode of replication. In semi-conservative mode, you know that the DNA, as you know, consists of two strands, that is, two helices of the original strand. They separate and they synthesize the complementary strand. That is how it replicates in a semi-conservative method. And this was experimentally shown by the experiment conducted by Messelson and Stel. You can see in the diagram that the parent strand separates and both they are forming their complementary strand. Messelson and Stel grew E. coli for many generations in a medium in which a heavy isotope of nitrogen, that is N15, had been substituted for the normal light isotope, that is N14. Means they first cultured the E. coli in a medium which was containing the 15N, that is the heavy isotope of nitrogen. This was the few bacterial cells were isolated, their DNA was isolated and purified, and when it was observed that 100% DNA was having the 15N DNA in them. After that, cells were allowed to grow and DNA was extracted and analyzed DNA isolated from the cell after one generation. That is, when you transferred from 15N to 14N and keeping it for about 30 minutes in the 14N DNA, and after that isolating the DNA and observing it, it was observed that 100% DNA was hybrid DNA. That is, one strand was of 15N and one strand was of 14N, and that is how it was referred as hybrid DNA, and the 0% was 15N DNA. After two generations of growing the E. coli in the 14N medium, again, that is after the interval of 60 minutes, when DNA was isolated from the E. coli, it was observed that out of the total, 50% was of lighter strand and 50% were of hybrid. You can see here when after two generations the E. coli was taken out, DNA isolated and purified, and it was observed that 50% DNA was of 14N and 50% was hybrid DNA. Then you will see in the next, that is in the third generation, after the gap of 90 minutes of transferring the E. coli from 15 to 14N, you will see that the proportionately 14N DNA has now 75% and the hybrid DNA is 25%. Here you must have observed that at subsequent generations, if you collect the DNA, you will find that there is a proportionate increase of 14N DNA and proportionate decrease of hybrid DNA. And through this, they succeeded in establishing that the DNA replicates in a semi-conservative method. Mode of DNA replication. It's a complex multi-step process involving many enzymes and protein factors, and it starts at a particular spot called as origin of replication. That is, in short, we call it as ORI. From here, the replication of DNA starts. Now look at this diagram. Here you will see this is the circled region is the origin of replication, from where the origin of replication starts. You have in this region single stranded DNA binding proteins. You have helicase enzyme, and of course you have the primase, which of course we'll be taking up just now. The replicating units of the chromosomes are called replicons. Replication starts with incision or a cut at a specific point, which is generally referred to as nick. This is brought about by the incision enzyme called as endonuclease. Activation of deoxyribonucleotides. You know that deoxyribonucleotides or deoxyribonucleotide monophosphate occur freely inside the nucleoplasm. There are 4 types. You know 4 bases are there in DNA. That is deoxy-AMP, that is deoxyadenosine monophosphate. Then deoxyguanosine monophosphate. Then you have deoxycytidine monophosphate. And then you have deoxythymidine monophosphate. Now, they are activated by enzyme phosphorylation, by the process phosphorylation, and this is catalyzed by the enzyme phosphorylase, and the phosphorylated nucleotides are deoxyadenosine triphosphate, that is DAPPP, then deoxyguanosine triphosphate, DGPPP, and then deoxycytidine triphosphate, and similarly you have deoxythymidine triphosphate, that is DTPPP. Now, DNA polymerase always accepts the nucleoside in the form of triphosphate, that is you have DAPPP, DTPPP, DGPPP, and DCPPP. You have DNA polymerase and of course the DNA template. So you get the newly formed DNA, that is DAP, DTP, DGP, and DCP, and of course 2 and pyrophosphates are liberated with tremendous amount of energy, and this is the equational representation that how the DNA molecule is formed. Exposure of the DNA strand. Topoisomerases form nicks and reseal the same in one strand of DNA helicases, unwind the helix, and the 2 strands are separated and forms a Y-shaped replication form. You can see in the diagram. In prokaryotes, these enzymes are replaced by DNA gyrases, helix destabilizing, that is HD, or DNA binding proteins. Now, in this diagram, you can say the replication fork is there. Then you have DNA polymerase enzyme. Then you have DNA ligase joining the short segment, which is there on the lagging strand or Okazaki segment, and DNA polymerase you have on the other, and that is the leading strand. This is how the Y-shaped fork, replication fork is formed. And remember, the DNA polymerase always synthesizes new DNA in the 5'3' direction, never in 3'5' direction. Now, RNA primer. It is necessary for the initiation of new DNA chain. RNA primer is a small strand of RNA which is synthesized at the 5th end of the new DNA strand with the help of primase enzyme. It is specific and forms primer on a free end of one strand and fork end of the other strand. Once addition of nucleotides, that is the DNA starts, RNA primer is removed and the gap is filled. Now you can see here the curved lines indicate the primers, and you know, on the lagging strand as well as on the leading strand, the primer is there. And this is formed with the help of enzyme R primase. Base pairing. The two separated DNA strands in the replication fork functions as template. Deoxyribonucleotide triphosphates come to lie opposite to the nitrogen bases of exposed DNA templates. This energy is used in establishing hydrogen bonds between the free nucleotides and nitrogen bases of the templates. Now you can see here that how these hydrogen bonds between guanine and cytosine, you will have three hydrogen bonds, and between thymine and adenine, you have two hydrogen bonds. That is how the base pairing takes place. Chain formation. It requires DNA polymerase 3, Kornberg 1957. It's a complex enzyme and has 7 subunits. In presence of magnesium ions, ATP, GTP is guanosine triphosphate, TPP, that is thymine pyrophosphate, and DNA polymerase 3. The adjacent nucleotide gets attached to the nitrogen bases of each DNA template by forming a phosphodiester bond. As replication proceeds rapidly from the place of origin towards the other end, new areas of DNA duplex unwind and separate because of the sequential opening of DNA double chain, and as replication to form two chains of DNA, replication is also called zipper duplication. DNA polymerase, as I told you earlier, can polymerize nucleotides only in 5'3' direction. Since the two DNA strands are anti-parallel, the two templates provide different ends for the replication. So replication in two strands proceed in opposite direction, because in one strand it will be in the 5'3' direction, other will be 3'5". Now look here, that is how the semi-conservative DNA replication is there. You can see the leading strand and the lagging strand, and these lagging strands are joined with the DNA ligase. And these lagging strands are also called as Okazaki fragments. And you can see the replication fork is there, opening the two strands which are used as template, and helicase is the enzyme which helps. One strand is formed continuously as the third end is open for elongation, so it is called as leading strand and can be built in 5'3' direction. You know that DNA polymerase always synthesizes in 5'3' direction. On the other strand, due to the exposure of a small stretch of template at one time, short DNA segments are synthesized, which is called as discontinuous, and it is referred as Okazaki fragment or lagging strand because it was first observed by Riji Okazaki in 1968. Each Okazaki fragment has 3000 to 4000 nucleotides. A RNA primer is required every time for the formation of Okazaki fragment, and after the removal of RNA primer, these Okazaki segments are together by the DNA ligase, as I have shown you in the figure. You can see here, again you have the DNA ligase, then DNA polymerase, you have the RNA primer is there, you can see the lagging strand and the leading strand. In lagging strand, these short stretches are joined with the DNA ligase, and then you have your helicase enzyme is there, and that is application for is there. Topoisomerase enzyme is also there. The proportionate increase of protein in DNA and proportionate decrease of hybrid DNA supports that DNA is replicating in a semi-conservative manner. I just told you earlier that if you go on isolating the DNA from the subsequent generation, you will see there is a proportionate increase of the protein in DNA and proportionate decrease of hybrid DNA. Semi-conservative mode of replication of bacterial chromosome was also demonstrated by J. Cranes using the sophisticated technique of autoradiography, and the material is first supplied with a suitable radioactive material tritiated thymidine, that is the H3. Heavy isotope of hydrogen replaces the normal hydrogen. During incarbonation of tritiated thymidine, autoradiograph prepared at regular known intervals, it would be expected that in the regions where one cycle of replication of DNA has been completed, density of dots will be higher than in the region where the replication has not taken place. Now look here. You have one diagram of autoradiograph where you will see where the replication has taken place. You will see the density of dots is more as compared where the replication has not taken place, and the other diagram is to show you the interpretation. Wherever you have two colors, it means the replication has taken place, and where you don't have the two colors, that the replication is not complete. So by observing the density of the dots, wherever it is higher, it is the region where the replication has taken place. Lighter density of dot is considered to indicate that only one of the two strands is labeled, while the heavier density of dot will indicate that both these strands are labeled. The generation time was worked out by the Cranes as 30 minutes in E. coli.